Hey guys, it's Storm. Today we're doing experiment 15. Isolation of chlorophyll and carotenoid pigments from spinach. Photosynthesis in plants takes place in organelles called the chloroplasts. Chloroplasts contain a number of colored compounds, pigments, that fall into two categories: chlorophylls and carotenoids. Carotenoids are yellow pigments that are involved in the photosynthetic process. The structures of A, alpha, and B, beta carotene are given in the essay preceding this experiment. In addition, chloroplasts contain several oxygen-containing derivatives of carotenoids, known as xanthophylls. In this experiment, you will extract, or I will extract, the chlorophyll and carotenoid pigments from spinach leaves using acetone as a solvent. The pigments will be separated by column chromatography using alumina as the absorbent. Increasing polar solvents will be used to elute the various compounds from the column. The colored fractions collected should be analyzed using thin line chromatography. It should be possible that you identify most pigments already discussed in your thin layer plates after development. Chlorophylls are green pigments that act as principal photoreceptor molecules of plants. They are capable of absorbing certain wavelengths of visible light and then converted by plants into energy. Two forms of these pigments are found in plants are chlorophyll A alpha and chlorophyll B beta. Two forms are identical except the methyl group that is shaded in the structural formula of chlorophyll A is replaced with the CHO group in chlorophyll B. Phenolphthalein A and phenolphthalein B are identical to chlorophyll A and chlorophyll B, respectively, except that in each case, the magnesium ion 2+ is replaced with 2 hydrogen ions. Alright guys, with all that said and that read, we're going to go ahead and start on part A of experiment 15. Now, because I don't have access to a centrifuge and I don't want to do a centrifugation, we're going to be using a different process for extracting them in part A. But then after we do that, we're going to do the same thing as written as part B and C in your books. So let's go ahead and get started with experiment 15, starting at part A, the extraction. Alright guys, I have about 0.25 grams of frozen spinach leaves in here, and I'm going to use this mortar and pestle, just smash it and get it into a nice little decompressed cake. I'll see y'all when I finish that. Alright guys, as you can see, I've beat into a nice little cake. You can see kind of all of those pigments on my mortar, on my pestle, on my thumb. Now I'm just going to add some acetone to turn it into a paste. Alright guys, I've added 5 milliliters of acetone and grinded it into a paste. It's more like a soup, really, a broth, but you can see I've gotten a lot of pigment into that acetone. So I'm going to do this maybe for a couple more minutes, just smashing and stirring. Then I'm going to transfer it to my Buckner funnel. Alright guys, we're going to go ahead and put this in the Buckner funnel. Okay, so I'm just going to set this down right there. Please don't roll. All of my pigments, I already wetted that filter paper with acetone. So now I'm just going to and then I'm going to add about 3 milliliters back into there. Rinse it around and filtrate again. Here we go. It's about 2. There's the other 1. And if you can see, I'm just going to try to get all of that those last bits and into my Buckner funnel. So now what I'm going to do, I'm going to scrape out that last bit with my spatula. I'm just going to scrape out that last bit, put it in there, and then rinse that with about 2 milliliters of acetone again, and then we'll move on. See you guys after. Alright guys, we're back. As you can see, that is the leaves I started with, right? The solids. You can see there's no color. There's no color left in them. They're colorless. I pulled out the pigments into my acetone. Okay, and sorry the clips are being shorter now, but I have to move quickly, right? Because acetone, highly volatile. So what I'm about to do is I'm going to take all that pigment, put it into this test tube, and I'm going to add 3 milliliters of water, 7 millimeters of hexanes. Quick change of plans. I added it to this instead because it frankly filled this up by itself. Alright, but now I'm going to add 3 milliliters of water. Boom. And then, 1 second, 7 milliliters of hexanes. There we go. 7 milliliters added into there. Plug it up. Now I'm just gonna shake it. I'll see you guys in a second. Alright guys, we're back. I took the stuff from here. After shaking it, put it into here. Now I'm just gonna shake this up. Make sure if you do this in person, you have to release the pressure on this. You can see right now I'm setting it with its top off because there is some real pressure that builds in there. I actually had to pay a pretty big fine when I took the class because I left this on there. You can ask Lisa. I left this on there and I went to ask Lisa something and then we walked back and it shot up out of there, hit the top of the hood and broke. So if you do it in person, make sure to release pressure, make sure never to set the cap on there. I'll be back after I finish shaking it and venting it like we did in the last experiment. Alright guys, we are back. I want to give you a close-up of this column we have. So you see this bottom fuzzy layer. Alright, that bottom fuzzy layer, that's our aqueous layer. Above it, let me see if I can hold this up. You see that clear part? Well, it's not clear. The green tinted, less opaque layer, that's our hexane. So what we're about to do is pull this bottom aqueous layer down into our funnel. I mean, just into here. It doesn't really matter where we're gonna get rid of it. But and keep our top hexane layer. And then we're gonna evaporate the hexane off, right? Just so we can see our pigments, make sure that we have some serious extracts in there. Whoa, focus. Thank you. And then we're gonna redissolve it in 0.5 milliliters of hexane, just to make sure we have it really nice and concentrated. And that's that. So I'll see you guys after I pull it out. Alright, this is the hexane layer after I pulled out the aqueous and poured this out the top. You can see it's a nice color of green. It's kind of a green yellow, which we'd expect since they're both chlorophylls in there and carotenoids. Yeah, so I'm gonna dry this to make sure there's no water. Focus, please. Oh yeah, make sure there's no water in there. And then I'm gonna evaporate off. Oh, extract it, put it into a clean Erlenmeyer. Evaporate off on my hexane to get the dry concentration, and then just half a milliliter of hexane. I have my drying agent in there, my anhydrous sodium sulfate. As you can see, moves freely, no clumping, it's nice and dry. Did a good job on my separatory funnel work. Now, just gonna transfer it into a clean Erlenmeyer with a pasture pipette and evaporate off on my hexane. Here we go. This is the drying agent. As you can see, there's probably some more in there that I could try to get out. Oh my goodness, I'm sorry. Can you see now? What if I do it like a beauty guru? Yeah, there we go. You can see there's probably some I can get out, but I really don't want sodium sulfate in this. It's really important I don't, so I'd rather, have that type of error than have sodium sulfate in my sample. Now this is my extract. To be sure I have pigments, I'm going to turn on my air, lower, here. and I'm going to evaporate off all this hexane. I'll see you guys, hold on, so you can see, just like that. I'll see you guys when this is done. Alright guys, here's what it looks like after I evaporate it all off. Now I'm just going to add half a milliliter of hexane back into this. There you go, there's a closer shot. I'm going to reach for my hexanes. I need a new pasture. And remember, 1 milliliter is 20 drops, so half a milliliter will be 10 drops. Just going to drop them around, try to get this all down at the bottom. I'll show you guys in a second. Alright, that's half a milliliter, maybe a little more, maybe a little less. But you can see I have all of my extracts concentrated right there. And that concludes part A. Now we move on to part B, our column. Yay. Hey guys, alright, this is part B. As you can see, I've set up my column. I would have showed you guys how to do it, but I hated doing it, and they would have been made for you anyway, so I didn't. Trust me, you didn't miss anything. So this top layer right here is a activated alumina. This little thing here is a cotton plug. That's pretty much it. It's just trying to get it into this tiny tube. Now I'm going to add 3 milliliters of hexane to it and make sure it doesn't go dry. And I'll come back to you guys. Alright guys, right now I'm running my hexane through my column. I think I made a mistake. Well, not a mistake, just an error. Like, I won't have to start over or anything, but if you can see, the rate of drops is so slow. And that's probably because I made my cotton plug at the bottom too tight. So it won't affect you guys. You'll get your results all the same, but I will be here for a while. But I'm just gonna let this get to the top of the alumina, close it up at the top, and then I'm gonna add half of my extract. Yep. Alright, see you guys when that reaches the top of the alumina. Slowly and painfully pulled out the hexane. You can see I have it sitting just at the top of the alumina. You see that? Now what I'm about to do is add 1/4 of a milliliter of pigment, just enough to have a little bit left to run my plate. I'm going to add it to the top and watch it seep into the top layer. Come back in a second. Alright, added my pigment. You can see it up there. Y'all see? And I'm just going to watch and see if I get a yellow band pulling down my column. Admittedly, my column is not very pretty. I don't know what happened, but we're just gonna keep going with this. You see I'm pulling out solvent into my test tube 1. See if I pull a yellow band out or not. I already added 1 milliliter of hexane. So if after that milliliter of hexane I don't start seeing a band move down, I'm going to go to a more polar solvent, 70-30 ratio of hexane and acetone. I'll keep y'all updated. So I've added 4 milliliters, let it run down, not getting a yellow band. Oh well. So what I'm going to do is I'm going to add about 4 milliliters of that, a little more polar, into here. Oops, let me stop. Oop, don't want to let it go dry, remember? So, as you can see, I have all my pigments still sitting up there, no yellow band. So what we're about to do is add a more polar solvent, see if we can get a band out. Alright guys, so I paused the column for a second. As you can see, I have a band running right there. Now I'm a little confused, and you guys tell me what you think. If you look, it's yellowish, bluish kind of. I'm gonna keep letting it run and see if I get a little more separation. But I think that's going to be my yellow band. I don't know. We'll see. Wish me luck. As you guys can see, I switched to test tube number 2. And I'm going to pull out this first band I have. I think I put too much solvent, but it said I had 4 milliliters. Anyway, I'm going to pull out that first band. Catch you guys after. Alright guys, we're back at it. You can see that yellow band is finishing up, and I got a tinted test tube. So it worked. Now you might say, Storm, that looks green. Yeah, I know. Or, I don't know. It doesn't look exactly like the textbook says it should, but it's there, and that's what counts. So I'm gonna wait till those drops run clear. You see those drops? Wait till they run clear, turn it off, cap it up, switch to test tube 3, and I'm gonna try to get a green, but something tells me it'll turn out more blue. I don't know, maybe not, but we'll see. I also have to get rid of all that, so wish me luck. Alright, it looks like I've pulled out all of the yellow pigment. As you can see, it's running clear, so I'm going to switch 2 out, 4, 3, wait for the rest of this hexane acetone mixture to run through, and then switch to straight acetone, and hopefully get out my green-blue band. Okay, so I'm pulling out the rest of that. Now I'm about to add just pure acetone in, which is a little more polar, and hopefully pull out my green and blue bands. Alright guys, this is the pure acetone. As you can see, it's not really pulling a band down. About that much is going through. I'm not feeling it, so when it finishes going through, I'm going to add acetone methanol 80-20 mix. Maybe that'll get my band moving. Alright guys, I don't know if you can see, but right here I'm getting a blue line coming down in the 80-20 acetone methanol mixture. So I'm just gonna wait for that to get to the very bottom, switch it out with test tube 4, and then be done. Alright guys, this is the green band I saw. I pulled it all the way out. Now I'm going to take the green band and the yellow band. I'm going to evaporate them. Alright guys, this is test tube 2 and 4. Yellow band, green band. And I'm just going to place them in this warm water bath. I'm just going to evaporate everything off, stopper them, and move to the TLC cream. Alright guys, this is test tube 4. So it is my green pigments. This is it evaporated. Come on. See, it's faint, but I see them there. That's all I really need. My yellow, same way, faint, but there. Now I'm going to move to part C, which is the TLC plate. Okay, this is the extract from the spinach. This is the yellow band evaporated, green band evaporated. I'm going to dissolve each one in a little bit of hexane acetone solution and then put them in my TLC plate. I have these 3 capillary tubes. I'm going to use it to take a sample of each and put it onto my plate. As you can see, I have my plate labeled extract, yellow extract, green extract, and I'm going to put it in this chamber. Alright, this is my plate after I added the extracts. I'm just going to place it inside this chamber and let it run. I'll let you guys watch me put it in. You can see, and it's just going to pull the hexane acetone mixture up the plate. Oh, you can see. You see how it's moving the bands? Man, it's running pretty fast. I'll let you guys know when it finishes. Alright guys, this is the results from the plate. You can't really see the green tint here, but just trust me, it's there. So you see the extract contained both the carotenoids and the chlorophyll here and here. And you can see those translated into the yellow and green. I think that's all the data you guys need. So I'll see you next week.